Hi guys, welcome back to Amalgamated Moi. I recently went down a taxonomy rabbit hole and then discovered something. Viral taxonomy. You see, viruses do not fit into our regular taxonomy system as they are not alive. I'll get into that later. So they need their own taxonomy system, which I will give you a hopefully brief explanation of. As I can't cover every species of virus, we will only be covering bits and pieces of each taxonomical level. With that, roll the intro. Before we get into taxonomy for viruses, let me explain regular taxonomy. Taxonomy is the categorization of living things based on their presumed relation to each other. We categorize creatures using this system: domain, kingdom, phylum, class, order, family, genus, and species. With each step narrowing down the relatives of the animal. Let's use the taxonomy of humans, beluga whales, and darkling beetles as an example. All three are in the domain of eukaryotes, meaning they are multicellular, the kingdom of animals, and our first split, beetles are in the phylum of arthropods, while whales and humans are chordates, meaning backboned. Continuing are insects and mammals, and the next split between whales and humans, with beetles, primates, and Artiodactyla, which includes animals like hippos, camels, and whales. Then the families of Tenebrionidae, Hominidae, and Monodontidae. That leads us to their genus and species, which are the final categories used to find a specific creature. For example, humans are in the genus Homo and the species Homo sapien. Now that you understand living taxonomy, let's move to viral taxonomy. Viruses don't fit this taxonomy tree because, as mentioned before, they are not alive. Now, it's easy to confuse bacteria and viruses, however, there is a difference. Bacteria fit into the taxonomy tree because they're alive. Viruses do not fit 3 necessary functions to be considered alive, so therefore they do not fit into the tree. 1. They are not cellular, and are rather simply genetic material, either RNA or DNA, with no cellular parts. 2. They cannot produce their own energy. And 3. They need a host cell to multiply, aka they can't breed on their own. Now that you understand why they can't fit into the pre-existing tree, let's look at the tree that they do fit into. The difference between living bacteria and non-living viruses was first categorized by Adolf Edward Mayer, who began by grouping infections inflicting tobacco plants. Later, in 1990, Francis Holmes suggested that the first layer should be based on the living organisms the virus infects: Phaginae for bacterium, Phytophaginae for plants, and Zoophaginae for animals. This idea for the group was later rejected as it did not use morphological differences to categorize the viruses. Today, the first layer is the realm. This has to do with the nucleic acids within the virus. There are 7 realms, and they are equivalent to the domains in the regular tree. These viral realms are for filamental DNA viruses, double-strand DNA viruses, single-strand DNA viruses, RNA viruses, satellite nucleic acid viruses, protein capsule viruses, and multi-protein capsule viruses. Realms are the only category where I'll be naming all the areas since there are far too many of the rest. So, if you're curious, go for a dive of your own, and if you find something interesting, let me know in the comments. After the realm, viruses follow the same order as living taxonomy, with kingdom, phylum, class, order, family, genus, and species. These are determined through morphology, where viruses are grouped based on the presence or absence of characteristics, whether that be proteins, chemical makeup, or methods of reproduction. There are over 3,600 species of virus, so let's look at a few famous ones and see where they split apart from each other on the tree and why. Let's use chickenpox, influenza alpha, the common cold, rabies, and COVID-19. We start with viruses as a whole, and our first split occurs with chickenpox being a DNA-based virus, where the rest are RNA-based viruses. The next split is at the phylum level, where COVID and the common cold are part of the phylum Pisciverotica versus rabies and flu, which are part of Negarovirotica. This split occurs from whether or not the virus's RNA strand can synthesize proteins independently, or whether it needs to use a host cell. Our next split is between the flu and rabies at the class level, due to whether or not their genomes are segmented. And the final split is at the order level between the common cold and COVID, where COVID-19 has a lipid envelope and the common cold does not. We continue through family, genus, and species, reaching the 5 exact viruses. Now, obviously, this is a simplified explanation, as explaining the entire functions of all viral anatomy would take an entire class rather than a short YouTube video. But this gives you the basic idea. Viral taxonomy is similar to living taxonomy, separating species into categories based on their anatomical makeup. Understanding viral taxonomy helps medical professionals in developing cures for these viruses, and helps them understand how one disease may be similar to another. But with that, that is all for today. I hope you guys enjoyed this video. Thank you all so much for watching, and I'll see you guys next time. Goodbye!